Hi everyone, I am Dr. C.S. Kulkarni, MD, and I will be teaching medical biochemistry. Before I start the topic, please like, share, and subscribe the channel. In the last session, we dealt with proteomics and enzymes. In today's session, we will deal with second group of coenzymes. These coenzymes take part in the reactions other than hydrogen in transferring the groups. The particular group or radical is transferred from one substrate to another substrate. Most of them belong to the group vitamin B complex group. ATP considered to be the energy currency in the body. Fiske and Supparov first isolated ATP in 1926. Lohmann in 1929 showed the importance of ATP in muscle contraction. In the ATP molecule, the second and third phosphates bonds are high energy bonds. During the oxidation of foodstuffs, energy is released, a part of which is stored as chemical energy in the form of ATP. The endergonic reactions are carried out with the help of energy released from hydrolysis of ATP. For example, glucose is converted to glucose 6 phosphate in the presence of enzyme hexokinase and ATP is converted to ATP. Metallo-enzymes. These are enzymes which require certain metal ions for their activity. In certain cases, example copper in tyrosinase, the metal is tightly bound with the enzyme. In other cases, even without the metal ion, the enzyme may be active, but when the metal ion is added, the activity is enhanced. They are called ion activated enzymes. Example, calcium ions will activate pancreatic lipase. Here is an example of certain metalloenzymes. Zinc is present in carbonic anhydrase, carboxypeptidase, alcohol dehydrogenase. Magnesium is present in hexokinase, phosphofructokinase, enolase, glucose 6-phosphatase. Manganese is present in phosphoglucomutase, hexokinase, enolase, and glycosyl transferase. Copper is present in tyrosinase, cytochrome oxidase, lysylase, lysyl oxidase, superoxide dismutase. Iron is present in cytochrome oxidase, catalase, peroxidase, xanthine oxidase. Calcium is present in lecitinase, lipase. Molybdenum is present in xanthine oxidase. Mode of action of enzymes. There are a few theories explaining the mechanism of action of enzymes. Each, perhaps each of them, tries to view the fact from different perspectives to explain a particular aspect of action. First, the lowering of activation energy. Enzymes lower the energy of activation. Activation energy is defined as the energy required to convert all molecules of a reacting substance from the ground state to the transition state. Substrates remaining in energy trough are to be placed at a higher energy level, whereupon spontaneous degradation can occur. Suppose we want to make a fire, even if we don't keep a flame, the wood will not burn initially, but we have to add kerosene or paper for initial burning. Similarly, activation energy is to be initially supplied. During the enzyme substrate binding, weak interactions between enzyme and substrate are optimized. This weak binding interaction when between enzyme and substrate provides the major driving force for the enzymatic catalysis. Enzymes reduce the magnitude of this activation energy. This can be compared to making a tunnel in a mountain so that the barrier could be lowered. For example, the activation energy for acid hydrolysis of sucrose is 26,000 calories per mole, while the activation energy is only 9000 calories per mole when hydrolyzed by sucrase. In this graph, we can see that Gibbs free energy is extrapolated against reaction progress, where activation energy of catalyzed reaction is much lower compared to activation energy of uncatalyzed reaction. Protonated form of histidine is an example of a general acid and its conjugate base, that is, the general base. The action of ribonuclease is an example of acid-base catalysis. Histidine residues 12 and 119 at the active site of a ribonuclease function as act as acid and base in catalysis. Histidine 12 acts as an acid and donates a proton to form the basic form. Then the 2' 3' cyclic phosphate is formed. Histidine 112 accepts a protein from the cyclic phosphatase and product is released. In this diagram, we can see that acid-base catalysis with the help of histidine. On the left side, protonated form or acidic form is present, whereas on the right side, non-protonated form or the basic form is present. In the enzymes of aspartyl protease family, catalysis involves two aspartyl residues which act as acid-base catalysts. Catalysis by pepsin, cathepsin, and protease of HIV, that is human immunodeficiency virus, belonging belong to this group of enzymes. Binding of substrate to preform site on the enzymes can induce strain in the substrate. The energy level of substrate is raised. The combination of a substrate strain and acid-base catalysis is seen in the action of lysozyme. The lysozyme substrate has a repeating hexasaccharide unit. Next is 3A, substrate strain. Binding of the substrate to the enzyme generates a strain conformation in the enzyme substrate complex. In the transition state, acid catalyzed hydrolysis of the glycosidic linkage by a glutamic acid residue at the active site generates a carbonium ion on the D residue. This relieves the energy generated in the initial enzyme substrate complex. This results in the transition state to the products. The glycosidic bond between the N-acetyl glucosamine and N-acetyl muramic acid on the bacterial cell wall is thus hydrolyzed. This accounts for bactericidal action of lysozyme. We can see in this diagram that on the left side N-acetyl glucosamine is present, whereas on the right side N-acetyl muramic acid is present. Coming to the next 3B, that is serine proteases. They are enzymes with the serine residue at the active site, and most of the proteolytic enzymes belong to this group. Examples are trypsin, chymotrypsin, clotting factors. Here in this diagram, we can see that the serine proteases, serine on the right side gives its hydrogen to histidine and thus forms a acidic serine form, whereas on the left side the aspartyl residue takes an hydrogen from the histidine residue and creates a basic form. Next example is C3, action of chymotrypsin. It is a combination of covalent and acid-base catalysis. The peptide bond is hydrolyzed through acid-catalyzed nucleophilic attack, utilizing a serine 195 residue of the enzyme. The enzyme complex substrate complex is formed by the binding of the aromatic amino acid residue from, with the hydrophilic pocket on the right on the active side of the enzyme. In this diagram, we can see that enzymes often combine with catalytic strategies, two catalytic strategies. First, general acid-base catalysis, and next is covalent catalysis. Active covalent acyl enzyme intermediate is formed. Cleavage of the peptide bond occurs. Serine 195, histidine 57, and aspartate 102 are the catalytic groups. Covalent catalysis. In the covalent catalysis, the nucleophilic, that is negatively charged, or electrophilic, that is positively charged group of the enzyme attacks the substrate. This results in covalent binding of the substrate to the enzyme. Similarly, coenzymes often form covalent bonds with the substrate. Here ends our session. Kindly like, share and subscribe the channel. Thank you.